All right, so this is Inheritance, a closer look. Part 2. So, here we're going to discuss the idea of multiple alleles. Normally we have the genotype of diploid organisms to contain 2 alleles. That's what always happens. However, at some loci, we can have multiple alleles within a group of organisms. So therefore, we have, a greater variety of genotypes and phenotypes. Genotypes that produce a greater amount of phenotypes. An example that I'm going to present to you is with the ABO blood group. So here we have several different genotypes. So there's alleles really. So IA, IB, and little I, where IA is dominant to little I, IB is dominant to little I, and IA is equal in dominance and therefore codominant to IB. So here's a little diagram. We have the phenotypes, which present the blood types, which is A, B, AB, and O. We have the genotypes. We have the antigens that they produce, and we have the antibodies to protect them from the foreign antigens. So our blood types, you're probably familiar with this already. We have A, B, AB, and O. We can get A with IA, or IA little I. We can get B with IB, or IB little I. We can get AB with IB, and we can get O with double little I. Now, the antigens that A produces, they produce antigens for A, B produces antigens for B, AB produces antigens for AB, but O doesn't produce any antigens. You're going to see later why this is important. Now, the antibodies that A produces are against B. The antibodies that B produces are against A. AB doesn't produce any antibodies since it has both. And you're going to see why this is important later on. And O produces against A and B. Now, the fact that type AB blood doesn't produce any antibodies makes it a universal recipient. Now that means that it can accept, if you're gonna donate blood the recipient can receive pretty much any blood type. They don't have to. You have to. The fact that the blood types produce antibodies means that they have to. When you have a transfusion, you have to be very careful that the blood that you're transfusing into that patient is of the same type. Otherwise, you could kill them because then they're going to be fighting against the blood that you're giving them. But with type AB blood, you don't have to worry about that. You can give them any blood type and it will match perfectly. Now, the inverse is true with type O. Since it doesn't produce any antigens, it's not going to be attacked by any antibodies, and therefore, that makes it a universal donor. I'm going to erase. Now let's continue our look into inheritance, a closer look into inheritance, by talking about gene interaction and doing a review of dihybrid crosses, which we talked about in Intro to Mendelian Genetics. So our first generation to P generation, which is the parental, we have, RR, big Y, crossing with little r, little y, little y. For our F1, big R, little y. Our F2 generation ends up with a 9:3:1 ratio. We'll end up with this, and we'll end up with this. Now, this occurs due to the law of independent assortment, which was one of Mendel's postulates. But genes don't always act independently in a phenotypic expression. Usually, they depend on genes at other loci, and this is due to interactions between genes at two loci or more. And this is what we call gene interaction. The fact that genes at different loci contribute to the determination of a single phenotypic characteristic. For example, since this is a dihybrid cross, at the beginning is one of them is all dominant, the other one is all recessive, and they produce one that's the F1 generation that's dominant, but it's big Y, little y, big C, little c. Cross them, and we get the F2 generation, where we have the 9:3:1 ratio, but something weird happens. The colors are different and the shapes are different. And that's because when gene interaction is involved, you have to determine the probabilities of single locus genotypes, and you have to multiply the probabilities of the genotypes, not the phenotypes. The phenotypes cannot be predicted without, taking into account the effects of the genotypes at the contributing loci. That's why we get all these strange colors out of it. Now here's another fun term, epistasis. Epistasis is a kind of simple concept. A lot of people get freaked out about it because later it gets a little bit more complicated with all the different types of epistasis. But epistasis is generally when one gene masks the effect of another gene at another locus. Now, this is different from dominance because dominance masks the recessive trait, but both the dominant and recessive allele are on the same locus of the gene, of the chromosome. So therefore, epistasis is different because it's affecting another locus. And the genes that are involved, we call them the epistatic and the hypostatic. Epistatic being the gene that is masking the other gene, the hypostatic gene. So epistatic masks hypostatic. Erasing again. Now, as for the different types of epistasis, I'm going to keep things simple for now. Maybe I'll do something more in depth at some other point in time about the different kinds of problem types and the ratios and how to interpret them. But for now, there's three kinds of epistasis: recessive, dominant, and duplicate. Duplicate recessive. Recessive is when you have two recessive alleles at a locus and they inhibit an allele at a different locus, and the ratio that you get is 9:3:4. Dominant is when one dominant allele inhibits expression of an allele at a different locus, and the ratio is 12:3:1. And duplicate recessive is when you have two recessive alleles at either of two different loci, and they can suppress a phenotype, and that's a 9:7 ratio.